Your entire body operates on a basic four-letter genetic code. Every cell, every tissue, and every organ relies on this precise sequence to function. But what happens when a single letter in that billions-long sequence fails? That microscopic typo cascades. A single wrong letter can cause systemic organ failure, chronic pain, and severe health consequences. This is the origin point of genetic diseases. To fix these errors, we are going to build a biomedical action map. We will trace the exact path from the physical origin of a disease deep inside the cell, straight to its engineered cure. Step 1 is isolating the exact DNA glitch. Step 2 is engineering a highly specific three-part biological tool to neutralize it. We cannot treat complex diseases until we map the exact molecular mechanics of the error. Once we have the map, we can engineer the biologic. We will start with how healthy DNA works. Specific sequences in your DNA act as blueprints, coding for the standard biological building blocks your body needs. For instance, a specific genetic sequence that reads CTT dictates the creation of healthy, round, red blood cells. This diagram illustrates a point mutation, replacing the middle T with an A. Moving down, that wrong letter forces the body to use valine instead of glutamic acid. At the bottom, that substituted amino acid warps the cell into a rigid sickle shape. Because these cells are no longer smooth and round, they physically jam together and block blood flow. This traffic jam deprives organs of oxygen, causing severe pain and structural damage. A single substituted letter in the genetic code produces cascading, system-wide health failures. Biological function is entirely dependent on structural integrity. So, how does the cell's internal machinery physically process and act on that flawed blueprint without realizing it is wrong? The answer lies in Crick's adapter hypothesis, which relies on a specialized molecule called tRNA to act as nature's blind translator. This schematic maps the translation mechanism. The bottom mRNA strand acts as a docking station. The central, fleshy tRNA adapter reads the sequence and locks into place, forming a vertical bridge. At the top, the adapter delivers a specific amino acid to build a protein chain. Now apply the mutation to this mechanism. If the mRNA docking station has a typo, the adapter is forced to deliver the wrong amino acid to the assembly line. The cell is flawlessly executing its instructions, but because it blindly translates a typo, a microscopic genetic error is converted into a physical disease. Because we now understand the exact mechanics of how cells process these errors, bioengineers can design customized biological tools to intervene and fix the problem. Diagnosing the genetic error is only the first phase. Now we move from mapping the problem to engineering the biologic solution. We can intervene using an antibody-drug conjugate, or ADC. This is a highly sophisticated three-part therapeutic tool built to hunt down very specific cellular targets. Here is an ADC structure. The foundation is the Y-shaped antibody, engineered to bind to specific antigens. Attached is a chemical linker, acting as a structural tether. Finally, the cytotoxic drug, the red star, is a toxic payload designed to destroy the target cell. The full ADC operates in a precise sequence. The antibody locates the specific sick cell, the linker ensures safe transit through the bloodstream, and the payload executes targeted cell death without harming surrounding tissue. For decades, treatments relied on generalized chemicals that destroyed healthy tissue alongside the sick. By engineering a biological delivery system that strikes only the intended target, ADCs eliminate that collateral damage. We have now charted the full action map. We started with a single microscopic typo in the DNA, traced how the cell translates that error into disease, and engineered a precise three-part biologic to neutralize it. Synthetic biology is actively using these blueprints today to build specialized treatments for aggressive cancers and genetic disorders that were previously considered untreatable, and the tools are getting sharper. Now that we can target specific errors, emerging gene editing technologies like CRISPR are allowing us to rewrite the genetic code itself. You cannot build a solution until you understand the mechanics of the problem. By mapping disease down to the molecular level, we now have the exact schematics needed to